Today we are excited to welcome two speakers, Doctors May Chakrabandhu and Edward Lemke. First up is Dr. May Chakrabandhu. May has an interdisciplinary background spanning food science, engineering, cancer research, biochemistry, and chemical biology. She earned her bachelor's degree in food science from KMIT in Thailand before being awarded the Fulbright Scholarship to pursue graduate studies at the Purdue University in the US, where she received her PhD in food process engineering in 2000. During her doctoral research, she integrated mathematics, physics, and biochemistry into developing computational models for aseptic food processing. The completion of the first draft of the Human Genome Project in 2000 inspired May to pivot toward biomedical research. She began her first postdoctoral research at the Ottawa Health Research Institute in Canada, focusing on protein-lipid and protein-protein interactions in human reproduction. In 2003, she joined the Institute of Signaling Developmental Biology and Cancer in Nice, France, where she was awarded the prestigious Komen Breast Cancer Foundation postdoctoral fellowship. Over the next 15 years, her research led to a new understanding of Pho signaling and led to the development of novel patient stratification methods for cancer treatment. In 2015, May became enamored with the power of genetic code expansion, a natural progression for all the best scientists, of course. Recognizing its potential, she established in 2019 a GCE facility at the Institute of Biology Valros, whose mission is to facilitate the widespread application of GCE technologies. Since building the center, May has worked with many national and international research teams to apply GCE to address key biological questions, and has also mentored postdocs, master's students, and PhD students in the use of GCE to explore new scientific ideas. May, it's a pleasure to have you here with us today. Please take it away.
Okay, thank you very much for your kind introduction. And let me share my screen here. So, and I would like to thank the GC for All Center for giving me the opportunity to share our work with you all today. So our facility actually has the aim to make GCE easily accessible to interested biologists. So I'll be talking to you about how we bring GCE to our colleagues that study neurodevelopmental disorder. So it all started when Michel Studer, an expert in brain development, approached me to see how we can use GCE to study the Bosch-Boonstra-Schaaf optic atrophy syndrome, or BBSOAS. It is a disorder that is caused by mutations in the NR2F1 gene. And basically, this NR2F1 protein is a nuclear receptor, is a transcription regulator. And Michel's team have described this protein as a timer that decides when and where and how many neurons would be made in the brain. And it is very important for the development of the eye and the optic nerves. So this BBSOAS syndrome is actually associated with so many symptoms, and it has many mutations that have been detected in the patients. And our question is, how do mutations contribute to the symptoms? And this is where it started. Most of the work on GCE that I will be presenting here today is done by Wan Chana, who was a wonderful PhD student that I had the opportunity to co-supervise along with Michel. And it was also supported by Michele, an awesome researcher in Michel's lab. And also later into the project, Paul brought his talent and joined us as well. And now he's also doing his PhD using GCE to study this disease as well. So I will be presenting to you some of the results that have already been published, and also some behind the scene unpublished content that I hope some of you might find useful. So many mutations and so many symptoms. We are interested to see how the mutations would impact the structure and function relationship. So actually we teamed up with Daniele and Valerio from the University of Verona. They are structural bioinformatics expert, because we think it would be nice if we could predict which mutations would impact the structure and function. So the thing is, the experimental structure of NR2F1 is not available, so they had to start out with building a good model for NR2F1. As we see here, NR2F1 has a DNA binding domain and the ligand binding domain. And these two domains have a lot of mutations associated with the BBSOAS. And in the ligand binding domain, there are several subdomains, and we believe that the mutations in these different subdomains would impact different protein-protein interactions, and that could be linked to the different sets of symptoms and severity. So we focus on studying the impact of mutations on the protein interactions. So our goal is to capture the protein partners at different interaction interfaces in NR2F1. And then we would like to do that in living human cells in physiological condition. And we would like to have as little perturbation to the protein function as possible. So classical techniques to study protein interactions are wonderful, and they have been very useful, but they also have limits. And this is where GCE comes in. With a lot of advantages, because we can incorporate photocrosslinking amino acids in NR2F1 to covalently stabilize the transient or weak interactions that we might miss if we use normal co-IP, for example. And we can also use it to look for partners in living cells. And we can also choose the photocross-linking positions on our protein. That's great. And we can also choose photocross-linking amino acids that have different properties, such as the distance for cross-linking and the target. So to incorporate photocross-linking amino acid in the cells, we have to choose several things. So we start with, for example, we have to, of course, choose where we want to put the amber codon, and so that we can incorporate the photocross-linker at the position that we want. We were interested mostly in the ligand binding domain, so we picked several positions in different subdomains. And we decided to use mammalian cells, the HEK293 T cells, to facilitate the screening and the test in the molecular levels. And we also, of course, have to choose the photocross-linking amino acids. And here are some of the examples of the common ones that are commercially available. And they all have different properties, but they all work under the biocompatible wavelength at the 365 nanometer. And also the synthetase and tRNA plasmids needed for incorporating all of these amino acids are generously made available by our colleagues at Addgene. So I would like to thank everybody here as well. And so for us, we are very concerned about the preserving the structure of the protein of our interest. So we are kind of leaning towards the photocross-linking amino acid that are a bit less bulky. And also we would like to capture the partners that bind very directly to the NR2F1. So to us, azidophenylalanine was our first choice, but just to be sure that we don't miss any good things, so we also tested other unnatural amino acids as well. It turns out that actually in the conditions that we would like to use, AZF actually have a higher incorporation efficiency as well. So for us, it was a good compromise. So we go for using azidophenylalanine. So with the choices settled, Vanshana turned into a GCE machine. He transpected, he cross-linked, he analyzed, do that all the way, repeat. So, but before we go to tackling the biological question, we have to look at the efficiency. We have to optimize our protocol and screen things. So for us, optimum conditions are actually the ones that would give us sufficient incorporation and enough of photocross-linked complex so that we can detect it easily. And also we would like to be able to use it further on in the downstream proteomics analysis. And the conditions that we want should preserve the cellular function. And very importantly, we would like to have high full length to truncated protein ratio, because it's not only the absolute amount of the protein, full length protein that we are looking at, NR2F1 that are truncated actually could be toxic. So we looked at different parameters. So for example, we screen the amber codon positions. So here you see the immunoblot of NR2F1 that has azidophenylalanine incorporated at different positions. And also this is done after a photocrosslinking. And as you can see here, this position, for example, has much better incorporation efficiency than these, for example. And you can also see here on this plot that different apparent sizes or molecular weights of the photocross-linked complexes. And this could reflect the true complex sizes or the difference in the conformations of the complexes. So we also tested different plasmids that are available on gene that we know can also incorporate azidophenylalanine. And in our hands, when we would like to do transient transfection to incorporate AZF into NR2F1, we found that the plasmid that is shared by Irene Cohen's lab worked best for us. And also we tested the ratio of the synthetase tRNA plasmids against to the target plasmid, and we found that actually when we have a higher synthetase to target plasmid ratio, things work way better for us. And this might be a bit different from several reports in the literature where actually lower synthetase to target ratio are used. So with all the conditions kind of settled, now we go about to check which sites on NR2F1 could capture suspected partners, like, for example, homodimerization partner or heterodimerization partners. So here's an example of the capture of the homodimer. So we do the co-immunoprecipitation of NR2F1 using flag tag, and we expect to have partners being detectable using the MYC antibody. And here you can see when we use a normal wild type NR2F1 without any photocrosslinker, it is very difficult to detect any dimerization partner. So, and this is a little bit of a surprise to us at the very beginning, because it was believed that this dimerization should be stable, but actually we don't have a real evidence at the protein level in living cells that dimerization occurs. So if dimerization does happen really in cells, it must be quite transient. Now let's look at what GCE gives us. So you can see here, once we put the photocrosslinker into NR2F1, we could capture the dimerization partner very efficiently, especially at this position. So now we are quite happy. Now we have the variant that can really capture the partner, but not too fast. We are not ready yet. We have to check first whether or not this variant is working functionally. So first of all, we check its localization, because wild type NR2F1 would localize fully almost in nucleus. And when you have the truncated form without the unnatural amino acid, it would be completely excluded from the nucleus. But once we incorporate the AZF, we found that actually it goes right back into the nucleus, just like the wild type, which is great. Then we look at the cell death function. So when you have the truncated form of NR2F1, it would increase the cell death when compared to the wild type. Once you have the full length with the AZF, it is coming back down to the same level as wild type, which is great also. Now the cell cycle. So when you have the truncated form, it would block proliferation. And once we have the wild type, the full length with the AZF incorporated, it comes back like the wild type. So we are very happy. And now we are confident to use this variant with AZF at this position to go and study the impact of mutations on protein-protein interaction. So now I'm going to show you some results that have already been published. So by using GCE, we were able to identify the mutants that would disrupt the dimerization of NR2F1, of different types of dimerizations. And independently, so from the structural bioinformatics, we were able to predict basically the same thing, that this same mutation would disrupt the stability and affinity of the dimerizations, all the pair of the dimerization that we study. So it is very encouraging to see that when we use GC, we were able to also validate the predictive models. So now I'm going to show you some results that are unpublished. So this is from when we tested our GCE procedure to see if we can use it for proteomics and to identify new partners. So we picked 3 different variants of NR2F1 that have the photocrosslinker at different interaction sites. And we did the IP of the complexes with and without photocross-linking, etc. And then we send it to the proteomics core facility at EMBL to identify the partners. And the results came back very encouraging. It's showing the power of GCE that allows us to site-specifically capture partners. As you can see here, you can see different sets of proteins that bind to different interaction sites. So for example, it all makes sense also that you can see co-activators and activators bound to the co-activator recognition site, for example. And one of the partners that came up is CRAP2. And this interaction is quite significant because it is suggesting that actually retinoic acid might be channeled to NR2F1 through this particular partner. And the predictive model that Valerio and Daniele did supports this hypothesis very well. And also we have seen the co-expression of this CRAP2 and NR2F1 proteins in mouse brain in the developing hippocampus and also in the ventral retina. And this is very important because it supports the relevance of this interaction in the hippocampal and the visual pathways, which is very relevant to the symptoms of BBSOAS. And now let's look at the effect of mutations on this interaction. So from docking simulation, Daniele and Valerio actually took the information that we got from GCE that the binding happens around this Q244 site. And then they did a prediction and give us the 2 mutations that would disrupt this interaction and 2 mutations that will increase it. But actually they didn't tell us. They want to keep things independent. So they wait for us to come up with our own conclusion. And lo and behold, we have the same thing. So the same kind of mutations would reduce and the other 2 mutations would increase the interaction. So this was very gratifying for all of us to see this kind of result. So by using our complementary methods, we were able to link mutations to structure and protein interaction and symptoms. And of all the mutants that we tested, all of them are associated to intellectual disability symptoms, but 2 mutants that are associated to visual impairments are the ones that disrupted CRAP2 and NR2F1 interaction and also destabilized the monomer. And the 2 mutations that are associated to seizure and autism spectrum disorder are the ones that increase CRAP2 interaction to NR2F1 and destabilize the dimer. So with this, we were able to show some genotype and phenotype relationship. And to conclude, GCE has allowed us to covalently capture elusive transient interactions and also to site-specifically identify new partners. And also it allows us to evaluate the effect of mutations on the protein-protein interactions in cells very efficiently. So by integrating GCE with structural bioinformatics, we were able to show the structural function relationship that is impacted by pathogenic mutations and also associate the symptoms to mutation. So this is important for diagnosis and treatment of this disease. And I would like to just end here by saying a big thank you to Michelle and her team once again, and also to Daniela and Valerio for going into this adventure with us. And I would like to thank the people at the GCE facility, Bruno for his support from the beginning, and also to the students and postdocs that came by into our facility through our wonderful collaboration. And also, I would like to thank my colleague Agnes at the flow cytometry facility. And also, I would like to thank Jenny and Frank at the proteomics core facility in the MBL. And a shout out for Irene Coyne for sharing her experiences in GCE and photocrosslinking and protein interaction. And I would like to thank my institute for seeing the power of GCE and support us. And also to thank the funding agency that make this work possible. And I would like to thank you all for your attention. So now I'm ready for any questions.
That was great, May. Thank you very much. While we're waiting for other people to ask questions, I have a couple. Effectively, when you're putting in a photocrosslinker, you're making a mutation at an interface of protein-protein interaction. So how much have you found, you want to be just outside the binding interface to get cross-linking? Or do you find that being in the binding site works better? And kind of what your thoughts are on this?
We try to not to be right smack in the middle of it. So actually, this is why we picked the 244 because it was close enough. It on top of that, it cross-linked very well. So it was like, fortunate.
Yeah. So
Yeah.
basically, it's doing some screening to find.
Yes, screening is the best thing to do.
And don't fall in love with one site.
No.
And then the other question I had was, how much did you find playing around with the actual UV intensity and duration affected your cross-linking? And is it different for different sites?
Actually, we found that what we use 15 minutes is way more than enough. And this is a common amount that I see people do. I think it might have some impact. We did 5 minutes. I could not say that, we do like, big systematic screen. We did 5 minutes, but, it might be a little bit too short. Yeah, so we go for 15 and the cells look fine. So we keep them like that. So just to be sure. And now you were talking about the sites, right? Is it different between sites? I could not actually say quite clearly because different sites also incorporate differently. So, I cannot say that, using different amount, the length of cross-linking time, would affect differently, in terms of different
Yeah, I didn't know
sites.
if you retroactively went back to positive cross-linking sites and then played around with UV conditions to say, okay, this one's a really good cross-linker. How low can we go? Because you want to go high to do a panning, right, to make sure you're getting it. But then, I don't know, because I have a history in cross-linking, and these are some of the issues that I had. But
so what was your issue?
Getting things to cross-link.
So now, which cross-linker are you using?
Oh, all of them. But regardless, you've gotten it to work, and it's really nice.
Yeah, we are just lucky, I guess.
I can ask a follow-up on similarly. I mean, classic question people always ask is which cross-linker works best, is more effective. Did you try any of the other cross-linkers that you mentioned, or did you just try the azidophy and it worked great and you moved with that one?
Actually, we tried just azidophy because actually we tried to incorporate the other ones and they don't incorporate as well. So, so we stick with this one.
Do you think that was more of a GCE problem rather than a cross-linking photophysics problem in that case?
Right.
Yeah.
Actually, we were concerned about that because we know that the one we picked is not on the top of the list in terms of cross-linking efficiency. But it did work enough already for us for now. But of course, we are interested to try some other ones as well because we know we missed some things for sure. But
right.
Yeah.
Cool.